In this lecture, we are going to look at the Bio 107 practical, that is the biology practical, BIO 107. All right, and we are going to look at food tests first to start with. Food tests, different types of food tests, and what they actually test for. All right, so we have the first one here. Now, these things you are meant to know them. This is not something you just memorize and then go for the exam. You are meant to know them. Is that okay? So we have the first food test here, which is Benedict's test. Now, Benedict's test is used to test for carbohydrate, specifically reducing sugars like glucose and maltose. You are going to know what reducing sugars are when you get to 200 level. Is that okay? But whenever you see anything on Benedict's test, know that it is used to test for what, my dear student? Carbohydrate. All right? It is used to test for carbohydrate. Benedict's test for carbohydrates. Now, the solution turns brick red orange precipitate. If you have a brick orange precipitate with Benedict reagent, are you getting it? Then that particular one, it means that that substance is a carbohydrate. What we said is the Benedict reagent comprised of, I don't want to mention the chemical because that one is much more advanced than what you should just know for this particular one. The only thing you should know is that we have what we call Benedict's test, and it is used to test for carbohydrates, specifically reducing sugars like glucose and maltose. And whenever you put the Benedict solution in that sample given to you, or the substance to be tested, if the color gives you a brick red or orange precipitate, know that carbohydrate is present. If it gives you a different precipitate, know that carbohydrate is not present. All right. The next one is iodine test. Now, my dear students, iodine test is used to test for starch. Is that okay? Iodine test is used to test for what? Starch. And the solution turns blue black. Iodine test is used to test for starch, and the solution turns what? Blue black. So if you put iodine plus any sample given to you, any substance given to you, and it turns blue black, know that starch is what? Present in that substance. Then Sudan 4 test is used to test for lipid. Sudan 4 test is used to test for what, my dear student? Lipid. Then the results you are going to see, if you put a particular substance and then after putting the Sudan 4 reagent, you have a red stain oil layer which separates at the top. You know that what is present there is what? Lipid. Is that okay? Then burette test is used to test for proteins, which turns solution violet purple. So if you put any substance in such a way that at the end of the day, you put, let's say this is a substance plus burette reagent, the reagent that is used, and the something turns violet, which is this color, the color of the pen, or purple, you know that what is present there is protein. Then we have the emulsion test for lipid. A milky white emulsion is formed. But the basic one, the one that actually came out that I want you guys to know, is Benedict's test. What is it used for? To test for carbohydrates or reducing sugars, which turn brick red orange precipitate. That is the basic one you should know. Then Sudan 4 test for lipid, and then lastly, burette test for proteins. When you get to 200 level, you are going to do all these tests in detail under, I think, biochemistry. All right? So that is that. Now, the next thing we are going to look at is microscope. Now, if I ask each and every one of us what a microscope is, we should be able to say something. What is a microscope? Now, a microscope is, of course, a laboratory instrument used to examine objects that are too small to be seen by the naked eye. It magnifies, it enlarges rather, substances that you cannot see ordinarily with your naked eye. It provides a magnified image of a small object. Is that okay? Now, let's look at the types of microscope. There are basically 2 types of microscope, my dear student. There are how many types of microscope? 2 types of microscope. Is that okay? We have the light microscope and the electron microscope. The light microscope are divided into 2. We have the simple microscope, then the compound microscope, and the stereo microscope. The simple microscope, that one have one single lens, just one lens. All right, that's a simple microscope. Any microscope that has one lens is a simple microscope. Simple, not single. Simple microscope have a single lens, just one lens. Are you getting it? Like your magnifying glass, those ones they are what? Simple microscope. Then we have compound microscope. This one has two lenses. We have the ocular and the objective lenses. These ones they are compound microscope. And the compound microscope, they are the basic ones that you find in the laboratory. The compound microscope are the ones you find more in the world, my dear student, in the laboratory. Those ones they are called compound microscope. Is that okay? The ones you find in the laboratory are called compound. Then we have the stereo microscope, which is also called the dissecting microscope. This one give a 3D view, like 3D view of the objects you are magnifying. Are you getting it? All these compound microscope, they will only give one side view, but stereo microscope will give a 3D view of everything you are magnifying. So these are the three types of light microscope. Can we go together? What are they, my dear student? We have simple microscope, compound microscope, and what? Stereo microscope. We said the simple microscope have what? Just a single lens. Compound microscope have two lenses, ocular lens and what? Objective lens. We are going to look at what these lenses are. The stereo microscope is also called dissecting microscope, and it gives a 3D view of an object, especially during dissection. Then we have the electron microscope, that is the other classification. Now, this classification, light microscope and electron microscope, are based on what is used for the entire procedure. Light microscope uses light, electron microscope uses what? Electron. We have two types of electron microscope. We have the transmission electron microscope, which transmits electrons through a very tiny piece of sample or substances that is to be enlarged. That one is transmission electron. While scanning electron microscope, it scans electron over the surface of what you want to magnify. That one is scanning electron microscope. So two types of electron microscope, my dear student. Transmission electron microscope, scanning electron microscope. Basic two types of microscope, light microscope, electron microscope. Under light microscope, we have simple microscope, compound microscope, or stereo microscope. Under light microscope, some persons also say we have the monocular, the biocular, and then we have the stereo microscope. All right. Then under electron, we have the transmission electron microscope and the scanning electron microscope. So these are the classification of microscope. Now, this is a microscope. One is binocular microscope, the other one is monocular. Why do we say this one is biocular and then this one is monocular? Biocular in the sense that it has two eyepieces. Monocular has just one what? Eyepiece. This one you use one eye to view. This one you use what? Your both eyes. Binocular. Are you getting it? Why this one is what? Monocular microscope. Is that okay? Now these are the various parts of the microscope. Although the labeling, you get the way the labeling there. All right? You get the way the labeling there. But let me point out the different parts for you. Of course, this part here is the eyepiece. Are you getting it? Then this part here is what they call the revolving nose piece. This part here, because this part is moving. Are you getting it? It's moving to give you different objective lenses. The objective lens are inside this place. They are inside this revolving nose piece. Then this part here, where you put your specimen in a slide, is what we call the stage. This part here like this, my dear student, is what we call the stage. Then of course we have the diaphragm, and then we have this part here is the part where the light comes from. All right, this is the part where the light comes from. That is that. Now we have things that help move the stage up and down. These ones here, this knob you see here, they help move the stage up and down. One is called coarse adjustment knob, and the other one is called fine adjustment knob. The bigger one is called coarse adjustment knob, and then the smaller one is called the fine adjustment knob rather. Then this back part here is the handle or the arm. This is where you hold your microscope. This arm here is where you hold your microscope. Is that okay? You carry the microscope and this arm here, and then you hold the other part, with your other hand, you hold the base, then hold the arm. These are the various type of microscope. Binocular, two eyepiece; monocular, one eyepiece. The same things that are here is the same things that are here. The only difference is that this one has two eye, while this one has one eye. Is that okay? So that is the various part of the microscope. Now let's look at the function of the various part. We have the eyepiece, which is the ocular lens. The lens at the top that you look through, it typically magnifies the image 10 times. This one that you look through here is the eyepiece. Are you getting it? This one here is the eyepiece. Is that okay? Now, objective lenses, the primary lenses that magnify the specimen, they are housed in a revolving nose piece. Common magnifications are 4x, 10x. The 4x, they are called scanning, 10x low power, 40x high power, and then 100x oil immersion. What do we mean by this objective lens? You see this place here, it's revolve, which we call the revolving nose piece. I've told you, the labeling is kind of wrong. Are you getting it? So this part is the revolving nose piece. Is that okay? Now, inside this revolving nose piece, you have the objective lenses. These objective lenses, you can see we have, this one we have 3. Some have different magnification. Each of the lens rather have different magnification. Some has 4x, one has 10x, the other one has what? 40x. And if there are 4, you have 100x. For this particular one here, there are 3. Are you getting it? This one you can only see 2 here. This is what we call the revolving nose piece. This is where the objective lens are housed. So you may be asked, the objective lens are housed here, my dear student. What's the correct answer? The revolving nose piece. Is that okay? So that is that for the revolving nose piece. The objective lenses. Are you getting it now? We said the eyepiece, the magnification that the eyepiece can give is 10x. So the eyepiece is what you view. It can give 10x. Let us assume there is no other lens, only the eyepiece. It will magnify the image like 10x. But now, objective lens, you know this one already magnified 10 times. Objective lens will magnify it more again, 40 times of this 10x. Are you getting it? Making the image more bigger and more clearer to view. Then we have the nose piece, the rotating part that holds the objective lenses. It allows the user to switch between different objective lens. The revolving nose piece, this part here, you can see it. These 3 things, you can move them. That's why they call them revolving. Are you getting it? You can move it. Maybe you want to use 4x, you move it to 4x, you view. If you want to use 10x, you move it to 10x, you view. If you want to use 40x, just like that. That's why they call it what? Revolving. It can move. That is what houses the what? The nose, the objective lenses. Then, of course, we have stage, the flat platform where the specimen are held. This place here is the what? Is your stage. All right? Then stage clips. Now, if you look at each of the stage, you will see something like a clip. Look at it. All these things you see here, they are clips that hold the slide in place to ensure that the something does not move anyhow. Those ones are called the stage clips. Is that okay? Now, the diaphragm, iris diaphragm. Now, the diaphragm is what controls the amount of light. Are you getting it? That passes through the specimen and into the what? Objective lens. Now, if you look at it very well, there's something I want to. I don't know if the diaphragm is very obvious here. Okay, this one. If I'm stretching my eye, these people can never see it at the end of the day. The diaphragm is this under here like this. Are you getting it? It's what controls the amount of light that we actually pass through here into what we have in the objective lens, the diaphragm. Now, it's more like attached to the stage. All right? Now, condenser lens. focuses the light from the illuminator onto the specimen. It is located below the stage. We have the condenser lens, although this one do not have all those condenser lens. Then we have the coarse adjustment knob. It moves the stage up and down significantly to bring specimen into general focus. This big part here, there's a big knob at the corner of this thing that you can rotate, you can twist it. That is called the coarse adjustment knob. Are you getting it? It moves the stage up and down. The lenses here don't move. Like, you cannot move the lens up and down. It is the stage you move. When you move the stage, the object will not be in focus. What helps the stage to move is the what? The coarse adjustment knob. To general focus, it uses only low power like 4x and 10x objective lens. Then the next one is the fine adjustment knob. Now, beside the coarse adjustment knob, you see a tiny knob here. That is called the fine adjustment knob. This one moves the stage slightly to sharpen and refine the focus details. So whatever the coarse adjustment knob are focused, to make it even more clearer, you use the fine adjustment knob. Then we have the light source, which is the illuminator. A light, often a mirror or electric lamp, that provides the light to illuminate the specimen. The light source is coming from somewhere, this place here, this thing. This is where the light comes from, travel through all these points, then up to the specimen. Are you getting it? Now, the arm is the curve, this is where it is held. All right, it is used to carry the microscope. You hold it with one arm and then carry it under. All right, so that is that. Then the base, all these one are story. Now the base is the bottom part of the microscope that supports and stability. It often houses the light source and electronic. So this bottom part here is the base. Then the body tube, the hollow tube that connects the ocular lens to the objective lens. It maintains proper distance between them. Where's the ocular lens? This one. Objective lens, this one. There's a tube in between that connects them. It's called the body tube. But the most important thing you should know is the light source, fine adjustment knob, coarse adjustment knob, the stage, where the specimen are kept is called the stage. Are you getting it? Where you view is called the what? The eyepiece. Is that okay? Please take note of it. Now, another thing you should always know is that carry a microscope with one hand on the arm and the other arm supporting the base. I've said that over and over again. Put one hand here, the arm, then the other arm should be on the base. You hold it like this. That is how to carry a microscope. Right? Don't just go and hold it like this. One hand must be on the arm and the other one at the base. Is that okay? Exactly. Now, let's look at diffusion, osmosis, and tonicity. Now, what is diffusion, my dear students? I believe each and every one of you know what diffusion is. Diffusion is the movement of molecules or particles from an area of higher concentration to an area of lower concentration. That is diffusion. This movement continues until the concentration is equal throughout the space. Let us assume you have something like this. The concentration of one substance is here. Maybe you mess. After some time, that mess will start spreading to this side. Are you getting it? Why some will be living here, spread. Now, the something will stop diffusing when the concentration at this part is equal to the concentration. This side is the right-hand side. All right, is equal to the concentration at the left-hand side. In that case, you say that your substance has finished diffusion. Now, in diffusion, molecules that can diffuse, gases can diffuse, even liquid, solid particles can diffuse, and so on and so forth. All right, these are substances that can diffuse. All right, now the next is osmosis. Osmosis is a special type of diffusion, particular to only solution or water, rather. Water and solution. Osmosis, particularly to only water and solution. Through a semi-permeable membrane from an area of lower solute concentration to an area of high solute concentration. That is what osmosis is, my dear student. So if you are asked, what is osmosis? Osmosis is a special type of diffusion which involves the net movement of water through a selectively permeable membrane. When you say a membrane is selectively permeable, it means that that membrane only allows certain things to pass through, but does not allow everything to pass through. It only allows certain things to pass through. It does not allow everything to pass through. Eh? That is when you say something is what selectively permeable. For instance, some of you only allow people who give you money to get into a relationship with you. You are selectively permeable. Those that don't give you money, what's the law? They irritate you. They say, John, not the bath. You know they spray perfume. Why? Because you feel John. So you are selectively permeable. You don't allow anybody to. Sometimes it's also maintaining your class. Not everybody should actually have access to you. For instance, not everybody have access to me. All right. Who is there? No. All right. So that is osmosis. Osmosis is a specific type of diffusion. It is the movement of water through a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration. So what happens is that water, let us say the solute concentration is here, is very high. Then this is water. The amount of water here is small. Solute concentration here is low. The amount of water here is much. What happens in diffusion is that because the solute concentration is low and water is much here, water will start moving from here to the side that the solute concentration is what? Is high. So it moves water from low solute concentration to high solute concentration. Are you getting it now? Water, it is the water that moves, not the solute. Move water from low solute concentration to what? To area of high solute concentration. So for instance, if I have 10 mole of sugar here, and here I have 2 mole of sugar, where do you think water will move to? Water will move towards where there is more sugar. Are you getting it? Because the sugar concentration here, in this case, our solute is sugar, is high. So water will always move. It is not the sugar that is moving, it's not the solute that is moving, it is water. That is why we call it what? Osmosis. That's why we call it what? Osmosis. So osmosis is the movement of water from a region of what? High solute concentration to a region of what? Low solute concentration. It is not the solute that move. I mean, from a region of low solute concentration to a region of high solute concentration. It is not the water, it is not the solute that move, rather it is water that moves across. All right? Through what? A semi-permeable membrane, a semi-permeable membrane, a semi-permeable membrane. The membrane must be what? Selectively permeable, or you can say semi-permeable. Selectively permeable. When we say something selectively permeable, it means it only allows certain substances to pass through. So it allows water to pass through, but it will not allow the solute to go. And you getting a selectively permeable membrane. Osmosis. Then let us now talk about tonicity. Now, tonicity is a term that describes the relative concentration of solutes outside of a cell compared to the inside of the cell. That is what tonicity is all about. It is a term that describes the relative concentration of solute outside as compared to the what? Inside. The inside of every cell, my dear student, we say is called the what? Intracellular space. The inside of every cell is called the ICF, the intracellular space, while the outside of every cell is called the ECF, or the extracellular space. Now, what we are trying to do, we want to compare the concentration of, let us say, sugar inside as compared to what? Outside. That is what tonicity actually does. How much is inside as compared to outside? If the amount of the solute inside is equal to the amount outside, there is a name for it. There is a what? A name for it. If the amount inside is equal to the one outside, there is a name for it. And there is something that happens too. If the amount on the inside is less than the amount on the outside, there is a name for it. If the amount on the outside is greater than the one inside, there is a name for it. Are you getting it? If they are equal, there is a name for it. And there is something that also happens. Now, tonicity, we could have a solution, or we could have a solution that is hypertonic, isotonic, and hypotonic. What do we mean when we solution is hypertonic? Now, most time when we are talking about tonicity, we are always talking about the one outside as compared to the one what inside. Are you getting it? We are always looking at the one outside as compared to the one what inside. When we say something is hypertonic, it means the one outside has a higher concentration, as opposed to the one what inside. When we say something is hypertonic, we say when we talk about tonicity, we always refer to extracellular. That means the ones outside of the cell as compared to the inside. So if we say it is hypertonic, we are saying the what? Outside is hypertonic. If we say it is isotonic, outside is isotonic. If we say it is hypotonic, outside is hypotonic. So what does each of these mean? What does each of these mean? For hypertonic, we say the concentration outside is higher than the one what? Inside. And according to osmosis, when the concentration, solute concentration is much outside as compared to inside, remember that we said that osmosis, the water will move from low solute concentration to what? High solute concentration. Remember, for hypertonic, it means outside is more, inside is low. So it means that water molecule will move from the outside, since inside is low, outside is high. Osmosis say move from low to what? High. So it will move from the inside to the what? Outside. And when water continuously moves out of something, what do you think will happen? That something will shrink. It will what? It will shrink. And that's the reason why you see this one is shrinking. Why? Because the water are moving out. You can see it. Water out. All this water, these are water. They are moving out. Are you getting it now? It will shrink. We say it is what? Hypertonic. So when we say hypertonic, means outside is more, have a higher solute concentration as compared to what? Inside. So water will flow from inside to outside, which will cause shrinkage of the cell. That is why you see this cell now start shrinking. All right. Now, the next one is isotonic. Isotonic is when the amount outside is equal to the amount what? Inside. You say it is what? Isotonic. When the amount outside is equal to the one what? Inside. You say it is what? Isotonic. Is that understood? You can see in isotonic, some are entry, so water are entry, some are going out. So there is no net change in concentration. Then for hypotonic is when the amount inside is high and the concentration of the solute outside is low. If the concentration of the solute outside is low and the one inside of the cell is high, we say such is what? Hypotonic. What do you think will happen? Extracellular fluid is low. That is why we call it hypo. Hypo means lower than expected. So extracellular fluid has a lower concentration as compared to the inside of the cell. So what do you think will happen? It means inside has more. According to osmosis, water will flow from where there is low to where there is more. So water will flow inside of the cell. What do you think will happen if water flow inside of the cell? It is something will swell. And if it is too much, before you notice, what will happen? It will burst. So that is what tonicity is all about. Hypertonic, isotonic, hypotonic. Hyper, outside is more than the inside. So water flow from inside outward. Iso, they are equal. There's no net movement of water. Hypo, the one inside, concentration inside is more than the one outside. So water will flow inside of the cell, which may swell and burst. All right. Now, what is the implication? Isotonic solution. We have it here. That a solution where the concentration of solute is equal to the concentration inside of the cell. There's no net movement of water the cell maintain the normal shape. We say such a solution is what? Isotonic. Then we have another one, hypertonic solution. A solution where the concentration of the solute is higher than the concentration inside of the cell. The concentration outside in the ECF is higher than inside. Then water moves out of the cell. The cell shrinks, which cause crenation in animal cell and plasmolysis in plant cell. Crenation in animal cell and what? Plasmolysis in plant cell. We say such solution is what? Hypertonic. Then we have hypotonic solution. A solution where the concentration of the solute is lower. That means the concentration outside the ECF is lower as compared to inside of the cell. So what will happen? Water will move from the ECF into the cell. The cell swells and bursts, which we call lysis in animal cell, and then it causes turgor pressure or turgid in plant cell. We say it is what? Hypotonic. So lysis is caused by hypotonic solution when it swells, then it bursts. Hypotonic. Why in plants, you now feel it gives it strength because it gives it what we call the turgor pressure, or become turgid, which acts as kind of skeleton in plant cells. Plasmolysis, the process where a plant loses water, we say in animal it's called crenation, causing the membrane to pull away from the cell wall. The cell shrivels and the plant withs. Hypertonic solution, higher solute concentration what? Outside. What causes plasmolysis is what? A hypertonic solution. The process where plant cell loses water. When the plant cell loses water, the cell membrane will pull away from the cell wall. Why? That is why cell membrane is called the plasma membrane. Plasmolysis, the plasma is broken down. Are you getting it? And a hypertonic solution, how they do it, they cause am. Is that okay? So you may be asked, what is plasmolysis and what causes it? It's a hypertonic solution, which will now cause water to move from inside of the cell, which will cause shrinkage, and at the end of the cell, the plant might, the animal cell might even die, or plant cell might die. Then we have hemolysis. Hemolysis is basically for red blood cell, more like the other one. Now in hemolysis, the process where a red blood cell swells and bursts due to an influx of water. What do you think will make a blood cell to swell and burst? Is it a hypertonic solution or hypo? Hypo will cause it to swell and burst. Are you getting it now? So the cell eruption releasing its content, hemoglobin. Hypotonic solution actually causes hemolysis. Hemolysis is, hem has to do with blood. Hemolysis generally have to do with red blood cell, RBC. Is that okay? Please take note of that. Very, very important. Very, very important.